Thank you very much, and it's a real pleasure to be here in front of you, and also for the ORIENTplus team to choose biology as one of the sciences that they show. I think 10 years ago, I'm not sure biology would have been top of their list, and it's really wonderful to be in this situation. So I'm going to give you a very quick kind of geeky crash course in molecular biology so that you will understand what I'm showing when I wave it up this time. So, as David mentioned, I'm Associate Director at the EBI. I've been involved in genomics since I was 19, actually. And interestingly enough, most people think I'm a computer scientist inside of the field of molecular biology. I'm actually a biochemist. So I'm a very geeky molecular biologist. EBI is in Hingston, South Cambridgeshire. For everybody outside of the UK, that means it's in Cambridge. And EBI is part of EMBL, the European Molecular Biology Laboratory, and that's a little bit like CERN for molecular biology. So it's an international treaty organization formed by European countries to further the research in molecular biology. So here's your very quick crash course so that you can follow the bit of science that I want to tell you about. So molecular biology is the study of how life works at a molecular level. We are machines. We are molecular nanomachines. It's a rather amazing thought when you think about it. And the components of the machine that we are from a variety of things. And I've given you a kind of a vague mapping to things that you would recognize in your computer. So DNA is a bit like the hard disk. It's rather inert. You read, you write occasionally, but you mainly read from it. RNA is a bit like RAM. It's very volatile. It's actually what DNA gets copied into most of the time. It's the kind of working stuff of the information transfer, and it actually does stuff as well. The business end of life, your muscles, your eye lens, the receptors in your membrane that make your neurons be able to fire so you can hear me, those are proteins. They form into these amazing protein three-dimensional structures. And there's kind of fuel and signaling molecules, and those are small molecules, small chemicals that power this. It's a bit like electricity. Biology has quite a lot of theories, but those theories are really theories of how things interact and how this came about, the theory of evolution. What there isn't is a theory that predicts the bases of the human genome. There isn't a theory that says these are the sequences of the human genome. We have to just go off and determine them. We have to read them, and then we have to store them in a database, and other people need to get them back out. And so very, very often, most of the time, we are determining worldwide in many different groups. I mean, certainly not at the EBI. This is the product of a huge number of research groups worldwide. And they're doing experiments to determine attributes of molecules, sometimes the molecules themselves. And then they put them into databases, and those databases are open and globally accessible. And so then other researchers take that information back out again to be reused. And so this globally accessible open databases are just a foundation of this. So I'm going to focus into my favorite molecule. I come from the DNA end of business, the hard disk end of molecular biology, if you like to put it that way. And this is DNA written out as a chemical structure. DNA is a chemical structure. It's a polymer. And it's a really remarkable polymer. When people first mashed up bits of life in the 1920s and the 1930s and tried to understand what was going on, this was not the molecule they thought was going to be carrying information. It looked far too simple. Proteins and other molecules looked very rich and interesting. This was thought to be a scaffold or something else. Now, as it happens, this polymer is made out of 4 different possible subunits, monomers. And I've written all 4 out here again in longhand in this chemical structure. But of course, because there's only 4 types, we give them 4 letters, A, T, G, and C. So although I could write down this thing as a chemical structure every time, we tend not to. We use this abbreviated form where we just write GATC, etc., etc., etc. And this is a long chemical molecule, and it's present in every one of your 3 trillion cells. It's really a remarkable thing when you think about it. And just for future reference, one of these letters, one monomer, is called a base pair, and we abbreviate that as BP. And in the DNA world, that's a bit like a byte. That is the fundamental unit thing that you're going to transfer. Now, back in the 1970s, this gentleman who actually ended up winning 2 Nobel Prizes, this was one of the Nobel Prizes he won for this, determined a way of reading this chemistry, this chemical molecule. Now, this is Fred Sanger, and at the start, you could only read 500 letters at a time. We have 3 billion letters in every cell. So it was a tour de force to read 500 letters of a polymer in a single experiment, but that was still a long way off from even working out one thing. Over time, this technology has improved, and this is one of the reasons why I think I'm on this stage now, and my colleague or my equivalent would not have been on the stage 10 years ago. So what I'm plotting here, or what NHGRI is plotting here, is the cost per megabase of DNA sequence, and it's a log scale on the y-axis. So it goes from $10,000 per megabase, and then it goes down to $1 per megabase. So notice the left-hand side here. And then on the x-axis is time. Just to orientate yourself, the white line is the equivalent reduction in cost of chips, the driver that we know for mobile phones, computers, and everything else, which is about an 18-month halving time or doubling time of chip capacity every 18 months. And of course, on a log plot, this is a straight line. So that straight line is an 18-month straight line doubling time. And you can see that DNA sequencing technology was tracking a very similar doubling time for a long time until a rather remarkable thing happened in 2007, when a series of new technologies came around in the DNA sequencing area. And suddenly, DNA sequencing improved, halved in price every 6 months. And that was amazing, and that has triggered this drop from tens of thousands per megabase to dollars, under $1 per megabase. And it pushes the cost of a human genome now for between $5,000 to $10,000, which is really quite affordable in many ways. My colleague, Dave Adams, who sequenced mice, he got a grant just before this curve. It has a consumables budget to sequence a series of important medical mice. The grant started just after that bump in this. And so, of course, his consumables budget was bigger than he thought. So he used half of his consumables budget to do his project. 6 months later, he looked at this, and he used half again of his consumables budget to do it again. If you think about it, apparently, he has an infinite consumables budget that will continue in the future. But what scientists haven't done is actually, in biology, what they haven't done is just done what they were going to do, but cheaper. What they've done, of course, is just really starburst out into the sciences. They've sequenced more humans than you might think. They've sequenced lots and lots of different species, and my colleagues from BGI will talk about one of the many species that are now being sequenced for this. So just to mention, just to put this in perspective, so every cell, we have 3 trillion cells, I think, roughly speaking, in our body, and each one of them has 2 copies of this polymer, which are 3 times 10 to the 9 long. It really is a remarkable, remarkable molecule. Please don't think that's the most important. The size is the most important. E. coli, it's been masked there, has about 10 to the 6. Yeast has also 10 to the 6. Medaka, which is in the Western world called the Japanese rice paddy fish, though it lives all on the eastern seaboard of China as well, which is my favorite fish, as it happens, has roughly speaking the same size as us. But there are many genomes that are much bigger than us, so don't consider the size of the genome to be kind of tremendously important. There's a big range of genome sizes out there. So back in the turn of the century, the Human Genome Project, which was based around the previous generation of sequencing technology, happened. And in effect, one person was sequenced. It was a mosaic of 24 individuals, but it was as if it was one. It was old school technologies, and it was a bit epic. And that's where I joined this science in about 1996. And it was a great and amazing time. Now, the Sanger Institute, which is on the same campus at the EBI, next door to the EBI, generates the same data volume as that project every 3 minutes. So that's not the case that you can go into the institute and give your DNA sample and 3 minutes later it comes back out again. It's a throughput issue. It still takes some time, but every 3 minutes that center generates the same amount of data as that single project. And so much of this has now shifted into the analysis zone, into the data manipulation, the data aspect. So I'm going to talk a little bit about the exciting things that many people around the world have done using this technology. So one of the things that we have learned as a big worldwide community, there are about, we estimate, at least over 100,000 different laboratories that do this work worldwide. And so one area was understanding the population genetics of humans. We are a rather remarkable species. We are a very genetically undiverse species. We're dominated by the genetics of the very small group of us that lived in East Africa. And we exploded out of East Africa in this tiny time frame and populated the rest of the world. And it's a remarkable story that's worth, there's all sorts of little sub-bits to this. For example, before we exploded out of East Africa, another hominid, Neanderthals, walked out of East Africa and was enjoying themselves in Europe and the Urals. When the group of people who left East Africa met the Neanderthals, they do what humans do. They had a party somewhere, presumably in the Levant or something like that. They met their new friends. And in fact, all the different groups who are outside of Africa, we all carry about 2% of Neanderthal DNA due to that initial party. I'll let you work out what happened during that party process. And so that's one story of many stories that have come through from understanding our genomes better. But we did that not to tell stories, but to associate traits with disease, genetic variants with disease. And so I'm showing on the left here, this is about HDL, that's good cholesterol. And you can see here there's a place on the genome which is either G or A. Now, in each one of you, of course, you have one copy from your mum and one copy from your dad. And so each of you are either GG, both mum and dad gave you a G, AA, both mum and dad gave you an A, or you're G and A. You got one G from your mum, one A from your dad. And in this case, this one place in the genome, you can see that the people who have AA have significantly higher HDL, good cholesterol. Now, I can do that for good, researchers can do that for good cholesterol, for bad cholesterol, LDL, for height, for hair color, for whether you have diabetes, for whether you have colorectal cancer, whether you have depression. All these different things can be measured and then correlated with these genetic variants. And this has become an incredibly powerful technique for us to explore disease. And on the right-hand side here, I'm showing an output of this. This is indeed myself, and I have been genotyped on the cheap. I have my genome sequenced on the cheap, which is called genotyping, by a company in California called 23andMe. And I'm slightly more at risk of colorectal cancer. So I have a slightly higher risk of that. Now, in fact, I can tell you now that doctors are deeply unimpressed by this because my weight, my age, whether my parents had colorectal cancer is far more informative than my genetics at the moment. But let me say very clearly that's the at the moment statement is very important here, and there are diseases which are progressing to the point where this information is becoming useful in the clinic, and that's particularly true for cancer. And I was involved in this project called ENCODE. It was an exciting project to understand how the genome works, and this is going to come onto this thing here. The DNA in our bodies are wrapped up in histones. And so the remarkable thing about DNA is that each one of our cells has a full copy of our genome. Our liver cell, our skin cell, our hair cells all have the same genome. However, there's obviously a big difference. A liver cell knows that it should be making these enzymes mainly to remove actually nasty chemicals from your blood and do all sorts of other things, whereas your hair cell knows that it's making keratin. So the same genome is being used in different ways. And a lot of that difference is encoded not in the DNA, but in the way the DNA wraps up around these protein molecules. And we know an awful lot about this, and we know more about this from this project. And this is called a histone, histone protein, and the DNA is wrapped around here. And Samir here will now give out some more of these. And the histone, have a little look, and there's a dengue virus coming around as well. So this is from our colleagues. David said, we think that 3D printing would be really impressive for this. Let me just show you here. If you look carefully on the side, you can see the DNA helix here wrapping around the side of this nucleosome. And this little tail here is where modifications get put. And that correlates and sometimes causes the difference between the liver cell and the hair cell, so that a gene is switched on in liver and switched off in the hair cell. And Samir or myself will be happy to give you a guided tour of DNA around histones when you like. So now I'd like to go to a different aspect of the use of genomics with my colleagues in BGI, and I'm not 100% sure how we swap over here to the video side. Does that work, Paul? So I hope Jin is listening to this. And these are slides from Jin. Sorry, Paul, I should have warned you. And Jin, I hope, is, or someone from BGI, is down in that bottom left-hand corner. Let me just introduce BGI. BGI is called the Beijing Genomics Institute. It is actually mainly based in Shenzhen, despite the name. It moved from Beijing to Shenzhen some, I don't know, 5 years ago, and it's become one of the biggest sequencing facilities in the world. It's probably the third or fourth biggest sequencing facility in the world, and it's made a big impact on genomic science from China. Okay, if we can't make it work, I can probably talk through this. Okay. It's not coming in. Okay, so let me just say this, the BGI, which, like I said, is based in Shenzhen. It has a number of other offices worldwide and other places in China, have been sequencing a remarkably diverse number of species. And this is a flower, the spider flower. It wouldn't surprise me if it's a Chinese flower. I'm looking at my Chinese colleagues to know whether it looks vaguely familiar. But clearly, the reason why they're interested is this is in this genus which has both C3 and C4 carbon metabolism. And if I can drag your minds back to early biology, if you remember, there are two fundamental ways that plants capture carbon from the atmosphere. One is called the C3 system. It's based around Rubisco. And the other one still involves Rubisco, but it involves shuttling carbon dioxide into a specialized cell, and that's called the C4 system. And if we could move more plants from C3 to C4, we would be able to make plants more efficient and capture more carbon. And so there's a real opportunity here in understanding the transfer between C3 and C4. And there's quite a lot of interest in the evolution. In fact, the C4 chemistry has evolved multiple times in the plant lineage. It wasn't a single time And so we have many, many examples. This is one example of a switch between C3 and C4 chemistry. Now I'm going to talk about, this is about the science, and I'm going to talk a little bit about this end of the business, the infrastructure part. So I don't need to tell this audience that infrastructures are critical. I don't think any of us, I did not wake up this morning and ring up the London Electricity Board and say, look, is electricity going to be on in London? Because if it's not going to be on, I don't think I'm going to come to this event. The video conferencing won't work. Nothing will happen. I think I'll just stay in bed. I made the assumption the electricity was going to run throughout today. We all make the assumption that the internet is going to work every time we pick up our mobile phone and do something interesting with it. We make the assumption that trains work. There's a slightly different prior to that, especially sometimes if you're in the UK and it's snowing. And so as infrastructure providers, we're only noticed when things go wrong. So only when things go wrong do we notice that a piece of infrastructure is not working. The bottom one here is Heathrow in the snow that has my colleagues in Finland just falling about in laughter because a small dusting of snow at Heathrow makes it completely stop, whereas Helsinki Airport just keeps on going. And for biology, we at the EBI feel that we're providing an infrastructure. Many, many researchers make the assumption that they can download the human genome and download the annotation of the human genome. They can download protein structures like this and do research on that. They don't worry about it. They don't say, oh, this may not happen. They assume that it's all going to work. And we want them to make that assumption. That's a very important assumption for researchers to make, for them to do exciting stuff. But we, the EBI, sit on a technical infrastructure. And this is just the 5 different machine rooms that the EBI runs between Cambridge, and in fact 3 of them are down in London. We have a slightly complicated network topology that allows us to deliver our services in a reliable way. But we have started to seriously use the network. This was last year. We have a 10 gigabit connection between us and our NREN Janet that then goes out to Géant and everywhere else. And you can see that that 10 gigabit connection occasionally now gets completely saturated. And this is normally when a new data set becomes available and the whole world wants to get that data set. But in genomics, I think it's, the distribution patterns between sciences are different, by the way. And we have a kind of bow tie structure of a smallish, relatively smallish number of centers feeding data in to 3 basic worldwide centers. And nearly always, they're slightly different place for different molecules, but they're nearly always placed in Europe, in America, and in Asia. And at the moment, and then those 3 centers synchronize, and then we all distribute back out again worldwide. So with colleagues such as the BGI in Shenzhen, the internet connectivity between us and China has been more challenging to deal with than the internet connectivity between, say, Europe and the West, between Europe and America. And it would be really, really great to be in a situation where we can have the reliability of the network that allows us to do things. Occasionally, disks get driven from Shenzhen to Hong Kong for transfer. And that is totally feasible, but it's not the best thing to happen. And occasionally, disks get put onto aeroplanes and get sent from one place to the other. And whenever that happens, I feel that we're not doing something right, and we should be using higher throughput networks. More and more and more, this is happening. It's a great thing, but we are at the situation where we see these little kind of bottlenecks, and I'm very excited about better internet connectivity across China. And just to say, this is a plot of the submissions from China into those 3 worldwide databases. So NCBI is in America, DDBJ is in Japan, and EBI is us here in Europe. And you can see that there's been an incredible growth of submissions from China. It's mainly due to the BGI Institute in Shenzhen, but also from other places, the Cass Institute in Shanghai and elsewhere. I regularly go to China now. I try to go there once a year. And I have a growing set of collaborators, not only in Shenzhen, but also in Shanghai, some in Beijing. It's mainly Shenzhen and Shanghai. And just a personal reflection on China. I think in the West, it's very easy to be over-focused on China's size and its recent economic growth. But what I have learned from my visits is that there's also an incredible depth of culture and academic tradition, actually, across these different sites of different sorts. And it's made me realize that there is a lot of opportunity in collaboration between China and Europe, and China and the West. And I really enjoy working with my collaborators. So this is the gardens in Suzhou, which was, I think, the old, or close to the old imperial capital in China before it moved to Beijing. This is the Chinese Academy of Sciences Max Planck Institute, Joint Institute in Shanghai, and this is a picture of the Cass Institute in Shanghai, people there. So with that, I'd like to finish. Do ask me some questions, and you can follow me on Twitter. And I blog, and in fact I've blogged twice about East-West relationships, if you're interested in following that up as well. So thank you very much for listening. I guess I can take any questions, or do you want to move on? 2 or 3 questions. 2 or 3. If anybody has any questions, feel free to ask. Okay, maybe not. All right. If anybody wants a guided tour of a protein structure, come to see me or Samir after.